The overall goal of this methods article is to introduce the detailed protocols utilized by our lab for studying the molecular recognition of double-stranded RNAs by chemically modified peptide nucleic acids.
This method can help answer key questions in the RNA chemical biology field, such as detection and targeting of RNA structures. The main advantage of this technique is that the designing principles can be applicable to the targeting of any RNA duplex structures. Demonstrating the procedure will be Desiree-Faye, a research associate from my laboratory.
To begin this procedure, add 4 microliters of a 35% glycerol solution to previously prepared RNA PNA samples and mix gently. Using a micropipette and gel loading tips, carefully add 20 microliters of each sample to a previously prepared polyacrylamide gel, making sure not to introduce any air bubbles. Run the gel at a constant voltage of 250 volts for 5 hours. After 5 hours, turn off the power supply and remove the glass plate from the gel stand. Then, remove the gel sealing tape and disassemble the glass plate. Gently remove the gel from the glass plate and place it in a container filled with 350 milliliters of deionized water. Carefully add 35 microliters of ethidium bromide to the container. And place it on a platform shaker on low speed for 30 minutes. After disposing of the ethidium bromide solution,